Good evening, everybody. সবাই এসে গেছো তো তোমরা? তাড়াতাড়ি জয়েন করো। সবার পার্টিসিপেশন দরকার। আজকে আমরা একটা ইম্পর্টেন্ট টপিক পড়ব। So let's start today's class. Biotechnology, Principles and Processes. One of the important chapter in our turn 2. So directly moving to our content. So in reduced syllabus, the genetic engineering are considered for turn 2. Recombinant DNA technology, that is rDNA. So here two chapters are there, chapter 11 and chapter 12. Today we will complete the chapter 11 and next day we complete the chapter number 12. So total 11 marks from these two chapters. So let's study. So starting our lesson, to pay respect to the eminent scientist Herbert Boyer. Western Pennsylvania, where railroads and mines were in density in most young men. He completed graduate work in University of Petersburg and postgraduated and studies in Yale. He performed studies on a couple of RE restricted enzymes of E. coli bacterium with specially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion. This is why they are called restriction enzymes, which left what has become known as sticky end on the strands. These clipped ends made pasting together pieces of DNA a precise exercise. The discovery in turn led to a rich and rewarding conversation in Hawaii with a Stanford scientist named Stanley Cohen. Cohen has been studying small ringlets of DNA called plasmids, and which float about freely in the cytoplasm in certain bacterial cells and replicate independently from the coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and then reinserting them to other cells. So combining the process with that of DNA splicing, you read this in the topic, in the inheritance chapter, what is DNA splicing, aimed at Boyer and Cohen to recombinize the segments of DNA in desired configuration and insert DNA in bacterial cells. which could then act as manufacturing plants for specific proteins. The breakthrough was the basis upon which the discipline of biotechnology was founded. So, two eminent scientists, Boyer and Cohen. Biotechnology that deals with techniques of using live organisms or enzymes from organisms to produce products and process full use to humans. In this sense, making curd, bread, wine, which are all microbe-mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organism, that is GMO, genetically modified organism, to achieve the same on a larger scale. Further, many other processes and techniques are introduced under biotechnology. For an example, in vitro fertilization leading to test tube baby, synthesizing a gene and using it developing a DNA vaccine, or correcting a defective gene, and all are part of biotechnology. EFB, very important, remember this one, the European Federation of Biotechnology has given a definition of biotechnology. Remember this one, that's very important. That encompasses both traditional view and modern molecular biotechnology. The definition is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Remember this one? That is the definition of biotechnology given by the European Federation of Biotechnology. The principles of biotechnology, among many, the two core techniques are enable birth of modern biotechnology. That is, first one is genetic engineering and second one is bioprocess engineering. Genetic engineering, the techniques to alter the chemistry of genetic material, DNA and RNA, to introduce these into host organisms and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of the sterile ambience in chemical engineering process to enable growth of only desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. So these are bioprocess engineering, and this one the genetic engineering. Remember the two core technique of biotechnology. The former provides opportunities for variation formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. In asexual reproduction, the genetic information that preserve the genetic information, so you can tell why these things are telling. Asexual reproduction preserve the genetic information. Can anybody tell this one? While the sexual reproduction permits variation, What's the difference between these two reproductive processes? That we say that asexual reproduction always preserves the genetic information. Anybody give this answer? Why we say this one? Yes, in asexual reproduction, single parent, no recombination, no crossing over. So, as usual, the genetic information are preserved. Very good, Ankita. While the sexual reproduction permits the variation, the traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering, which include the creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. So very important paragraph. This is the core component of this chapter. The technique of genetic engineering include the creation of rDNA and the gene cloning, and from there the gene are getting transferred. So in this way it overcomes the limitation and it is always isolate one or the desired genes, a set of desired genes, and without introducing the undesirable genes into the target organism. Remember this one, that's very important. Most likely the piece of DNA would not be able to multiply itself in the progeny cell of an organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along the host DNA. So that's very important. This is a key point in biotechnology. That is a question also. Why the piece of DNA is not able to multiply itself? So, and the next part is the answer. When it is get integrated into the genome of the recipient, it multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA sequence called ORI, origin of replication, which is responsible for initiating replication. Another important thing, initiation of replication done by ORI, the origin of replication, the specific DNA sequence that is the initiating all the replication called origin of replication or ORI. Therefore, for the multiplication of any alien piece of DNA of an organism, it needs to be a part of the chromosome or chromosomes which has a specific sequence of origin of replication. Very important thing. Thus, an alien DNA always linked with ORI, origin of replication, so that this alien piece of DNA can replicate and multiply itself in the host organism. So, alien DNA always linked with ORI for starting the replication. This can also be called as cloning or making multiple identical copies of any template DNA. So everybody understood this one? So moving to our next part, the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with native plasmid, Salmonella typhimurium. So that's what is plasmid, that is written in the bracket. Autonomously replicating circular extra chromosomal DNA is called the plasmid. So that's the important sentence to remember what is plasmid. Previously we discussed the two eminent scientists Cohen and Bayer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting a piece of DNA from a plasmid, which are responsible for conferring antibiotic resistance. The cutting of DNA at specific locations become possible with the discovery of molecular scissor. Very important, you all know this one, the molecular scissor, RE, restriction enzymes. The cut piece of DNA and then linked with the plasmid DNA. This plasmid DNA acts as vectors to transfer the piece of DNA attached to it. You probably know that the mosquito acts as insect vector to transfer the malarial parasite into the human body. In the same way, the plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. So what is the function of plasmid? Is to deliver an alien piece of DNA into the host body. The linking of antibiotic resistant gene with the plasmid vector become possible with the enzyme, another enzyme, you know this one, that DNA ligase, that is the molecular glue, which cuts the DNA molecule and joins their ends. This makes a new combination of circular autonomously replicating DNA created in vitro known as rDNA or recombinant DNA. So when the piece of DNA is attached by the help of plasmid vector and attached with the DNA by DNA ligase, so this new combination is called the rDNA. So this new combination is called the recombinant DNA. When the DNA is transferred into E. coli, a bacterium closely related to Salmonella, it could replicate using a new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant genes of E. coli is called cloning of antibiotic resistant gene in E. coli. So you can hence infer that. So there are three basic steps of genetically modifying an organism. Very important question, you can say. Identification of DNA with desirable genes, introduction of identified DNA into the host, maintenance of introduced DNA in the host, and transfer the DNA to its progeny. So these are the three basic steps in genetically modified organism: identification, introduction, and maintenance. So next part of our lesson, the tools of recombinant DNA technology. First started with the RE, the restriction enzymes. In 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added methyl groups to DNA, while other cut DNA. The latter was called, which was cut the DNA, that is called the restriction endonuclease. And the first RE, that is HIN2. I think you all know the naming process of RE. The H came from the genus first name, that is Haemophilus. Next two letters came from the species two letter Influenzae, and D is the strain and 2 is the series number. So HIN2, whose functioning depend on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIN2 always cut DNA molecules at a particular point by recognizing specific sequence of 6 base pairs. The specific base sequence is known as called the recognition sequence for RE HIN2. So these are all information, keep it into mind. The 6 base pairs in the first case, and this is called the recognition sequence, in which point are cut by the restriction enzyme HIN2. Nowadays, lot of RE are developed. See the fact that more than 900 RE has been isolated from 230 strains of bacteria, each of which recognize different recognition sequences. So keep it in mind, all the information, that is very important. Everybody understood this one? So everyone give a response and like and share our channel and participate. So you can call your friends to participate our lesson. Those are giving term to exam. All are benefited. The convention for naming these enzymes that I told the earlier slide, the first letter of the name comes from the genus, second two letters come from the species of the prokaryotic cell from which it was isolated, such as E stands from Escherichia, CO stands from coli, and R, that is the strain number, and this is the first series. R is derived from the name of the strain, the Roman number following the names indicated the order in which the enzyme were isolated from the strain of bacteria. Restriction enzyme belong to a larger class of enzyme called nucleases. These are two kinds, exonucleases and endonucleases. The nucleases are bifurcated into two. Exonucleases remove the nucleotide from the end. That means when they remove from the end part, they cut from the end part, these are called exonucleases. Whereas endonucleases make cut at the specific position, so they cut from a specific position of DNA within the DNA, that is within DNA, and that is from the ends of DNA. So this is the ends of DNA and that is the within DNA, and one is exo and another is endo. Each restriction endonuclease function by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of a double helix at a specific point in the sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. You can remember, anybody give example of the palindromic sequence? We discussed this topic in many times. What is palindromes and what is the palindromic sequence? Can anybody tell me? Okay, the enzyme cuts both DNA strands at the same site. So that is the, here the diagram is shown. The EcoR1 cut DNA between the bases of G and A only when the sequence GAATTC represents in the DNA. So this is the sticky end. This is the sticky end. And DNA fragment join at a sticky ends. So these components. And now it is a, when the foreign DNA is going to attach in the sticky ends, now it's a rDNA or recombinant DNA. Here you see the vector DNA, that is the foreign DNA. They cut in a particular place, that is G and A sequence. Now this one attached with they form sticky end and they are attached. See the A attached with this one. So everyone AT, GC, this type of bonding are there. And form the recombinant DNA. So here the description of the palindromes. These are group of letters that form the same word when read both forward and backward. See the word Malayalam. From the forward and backward, you can see the same word. As against a word palindrome, where the same word is read in both directions, the palindrome in DNA in sequence of base pair that reads same on two stands, when orientation of reading is kept the same. See the example here. One from 5 prime to 3 prime and another is from 3 prime to 5 prime, that is GAATTC, and from 3 prime to 5 prime, that is CTTAAG. That's the same direction in both from 5 prime to 3 prime and from 3 prime to 5 prime. That is the palindromic sequence. Restriction enzyme cut the strand of DNA a little away from the center of the palindromic sites, but between the two bases, the same two bases, on opposite strands. This leaves single-stranded portion at the ends. There are overhanging stretches called streaky end on each strand. These are named so because they form hydrogen bonds with the complementary cut counterparts. This stickiness ends the facilities of action the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources and genomes. When cut by the same restriction enzyme, the result of DNA fragment have been same kind of sticky ends that are joined together using DNA ligase. Here you see the foreign DNA. Same restriction enzyme cut both the foreign DNA and vector DNA at specific point. And ligases join the foreign DNA to the plasmid. This is the place. See the rDNA technology high, recombinant DNA molecules are formed. Now this goes for transformation. This is the E. coli, and now the cell divide. So this is the flow chart of rDNA technology. So take a screenshot. So everybody take the screenshot. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease result the fragments of DNA. These fragments can be separated by the technique, very important, gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them move towards the anode under the electric field through a medium or matrix. Very important concept. Nowadays, the most commonly used matrix, the name of the matrix is agarose, which is a natural polymer and extracted from the seaweeds. The DNA fragment separate according to their size through sieving effect provided by agarose gel. Hence, the smaller the fragment size, the further it moves. Very important concept. Now the separate DNA fragment by gel electrophoresis can be visualized only after staining the DNA with ethidium bromide. Very important. Frequently asked question. Ethidium bromide followed by exposure to UV radiation. You can see the bright orange color blends of DNA in ethidium bromide strand gel exposed to UV light. The separated band of DNA are cut off from the agarose gel and extracted from gel piece. This step is called elution. Very important term. What is elution? Keep it into mind, that's very important. So the separate bands of DNA are cut from the agarose gel, extracted from the gel piece. This step is known elution. The DNA fragment purified in this way and used by constructing the DNA by joining them with cloning vector. See the diagram that is also given in your question. You can get this question in your board exam. The cathode and anode are there. See, the smallest part are moved towards anode and the largest parts are moved in the cathode. A typical agarose gel electrophoresis is shown in the fragment. Set of DNA fragments are there. Now the cloning vectors. You know the plasmids and bacteriophages have the ability to replicate within the bacterial cell independently or control the chromosomal DNA. Bacteriophages, their high number per cell, have the high copy numbers of the genome within the bacterial cells. Some plasmids may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. So their number can go even higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. So vector used at present are engineered in such a way that they help linking of foreign DNA and selection of recombinants from non-recombinants. See the one of the important part of our lesson, PBR 322. So everybody remember this one, the location of this RE. E. coli cloning vector PBR 322. The restriction sites are there. So hint 3, Eco R1, BUM H1, Cell 1, PVU1, PST1, PVU2 and CLA1. This is the origin of replication. And the replication of the plasmid is there. So these are the selectable marker, ampicillin resistance and TET-R both are there. So everybody keep in mind the diagram for further practice. That is one of the important question in biotechnology. So make the drawing. So take a screenshot and remember all the restriction enzymes that are present on PBR 322. So moving to the next part. So the following are the features that are required to facilitate the cloning into vector. ORI, origin of replication, we know this one. This is a sequence from where the replication starts, and any piece of DNA when linked to the sequence can made to replicate within the host cell. The sequence is also responsible for controlling the copy number of the linked DNA. So, if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. Another important part, that is the selectable marker. The vector requires a selectable marker which helps to identifying and eliminating non-transformants and selectively permitting the growth of transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin, etc. are considered the selectable marker for E. coli. So in PBR 322, ampicillin and tetracycline markers are there. E. coli do not carry resistance against any of these antibiotics. The cloning sites, in order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used RE. Presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out a restriction site present in one of the two antibiotic resistant genes. For example, you can ligate a foreign DNA at the BAMH1 site of tetracycline resistant gene in vector PBR322. I think you all know the name: P for plasmid, B for Bolivar, R for Rodriguez, and this is the lap number. The recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but can still be selected out of the non-recombinant ones by plating the transformants on tetracycline containing medium. The transformants growing on ampicillin containing medium and are transformed and are transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on the containing tetracycline. But non-recombinant will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistant gene get inactivated due to insertion of alien DNA and helps the selection of recombinants. Selection of recombinant due to inactivation of antibiotics is a process because it requires simultaneous plating of two plates having different antibiotics. Therefore, alternative selective markers have been developed which determine recombinant from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. The recombinant DNA is inserted with the cloning sequence enzyme that is known as beta-galactosidase. This results into inactivation of the gene for the enzyme, which are referred to be insertional inactivation. Very important, insertional inactivation. That is the inactivation of gene synthesis of this enzyme. The presence of chromogenic substrate gives blue colored colonies if plasmid in the bacteria do not have any insert. Presence of insert result into insertional inactivation of beta-galactosidase gene, and the colonies do not produce any color. This identified as recombinant colony. So the chromogenic substance gives blue colored colonies when the plasmid of the bacteria in the bacteria does not an insert, and presence of insert do not produce any color that identified recombinant colonies. So everybody understood this one? So vectors for cloning genes in plants and animals. You may not be surprised to know that we have learnt a lesson from transferring genes into plants and animals from bacteria and viruses, which have known for ages how to deliver gene to transform eukaryotic cells and force them to do what the bacteria or viruses want. Agrobacterium tumeficans, a pathogen in several indicot plants, is able to deliver a piece of DNA called tDNA to transform normal plant cells into tumor and direct this tumor cell to produce a chemical required by the pathogen. Similarly, retroviruses of animals have the ability to transform normal cell into cancerous cell. So a better understanding of the art of delivering genes by pathogen in their eukaryotic host has generated knowledge to transform their tools of pathogen into useful vectors of delivering gene to insert to human. The TI plasmid, that is the Agrobacterium tumeficans, has now been modified into cloning vector, which is no more pathogenic to the plants but still able to use the mechanism to deliver gene of our interest to verify plants. Very important paragraph to remember. It's a modified into cloning vector without pathogenic activity. Retrovirus is also been disarmed and now used to deliver desirable genes into the animal cell. That is a new activity in biotechnology. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred to a bacterial, plant or animal host where it multiplies. So the competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, remember this one, it cannot pass through cell membrane. Why? In order to force the bacteria to take up the plasmid, the bacterial cell must first be competent to take up DNA. This is done by treating them with a specific concentration of divalent cation such as calcium, which increases the efficiency with that DNA enters the bacterium through pores in the cell wall. Recombinant DNA can also be forced into such cell by incubating these cells with rDNA on ice, followed by placing them into (42°C). That is 42°C. And then putting them back on ice. This enables the bacterium to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cell. In a method known as microinjection, recombinant DNA is directly injected to the nucleus of an animal cell. In another method suitable for plants, cells are bombarded with high velocity of microparticle of gold or tungsten coated with the DNA. That is called gene gun or biolistic method. So one is microinjection, another is biolistic or gene gun method. And the last method uses disarmed pathogen vector, which when allowed to infect the cell, transfer the recombinant DNA into the host. Now we have learnt about the tools of constructing rDNA. Let us discuss the process of facilitating rDNA technology. So here the process of recombinant DNA technology, that is our next topic. So very few are listening. Everybody come first and give your presence in biotechnology lesson. So recombinant DNA technology involves several steps in specific sequence, such as isolation of DNA, fragmentation of DNA by RE, isolation of desired DNA fragment, ligation of DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extracting the desired product. So these are the steps that are taken in rDNA technology. So first, the isolation of the genetic material of DNA, that is DNA. Recall of nucleic acid recall that nucleic acid of the genetic material of all organisms without exception. In majority of organisms, that is deoxyribonucleic acid, in order to cut the DNA with RE, it needs to be pure form, free from microorganism. That is the isolation. Since DNA is enclosed within membranes, we have to break the cell open to release the DNA along with the macromolecules such as RNA, protein, polysaccharide, and also lipids. This can be achieved by treating the bacterial cell. The enzymes are used. First one, number 1, lysozyme in bacteria. Number 2, cellulase in plant cell. Number 3, chitinase in fungus. Very important, we know this one. By the activity of the bacteriophage, the bacteria, inside the bacteria, the virus inserted its DNA by secreting the enzyme lysozyme. Cellulose dissolving enzyme, that is cellulase, and chitin dissolving enzyme, the chitinase, in fungus are used. You know that the genes are located on long molecules of DNA intertwined with proteins and histones. The RNA can be removed by the treatment of, very important, ribonuclease, whereas the proteins can be removed by the treatment with protease. One molecule can be removed by appropriate treatment and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen in collection of fine threads in the suspension. So here the DNA is separated out, can be removed by spooling. That's very important question. So cutting of DNA at specific location by RE, the purified DNA molecule by RE in the optimal condition in a specific enzyme, agarose gel electrophoresis, check the progression of the restriction enzyme, digestion. DNA is negatively charged, we already discussed this one, hence it moves toward the positive end, that is anode. We already discussed this point. Very important point, that DNA is always negatively charged and it always moves towards the positive electrode, and that is anode. The process is repeated with the vector DNA. The joining of DNA involves several processes. After having cut the DNA, the source of DNA as well as the vector DNA with the specific RE, the cut out the gene of interest, that is the desired gene, from the source DNA, and the cut vector with the space and mixed with ligase is added. This results the preparation of the recombinant DNA by using the ligase. After this, the amplification of the gene, that is one of the vital question in our turn to what is PCR and why it is used. So everybody very keenly observe this slide. The PCR, the polymerase chain reaction, that's one of the important question for your coming exam. In this reaction, multiple copies of genes in DNA are synthesized in vitro using two sets of primers. Small chemically synthesized oligonucleotides that are complementary to the regions of DNA and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If this process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately 1 billion copies are made. Such repeated amplification is achieved by the thermostable DNA polymerase isolated from the bacterium. Very important question, Thermus acuitas. The thermostable DNA polymerase isolated from Thermus acuitas, which remain active during high temperature, introduced by denaturation of dsDNA. The amplified fragment, if desired, can now be used to ligate with the vector for further cloning. So we are moving to the diagram of PCR. From there we can revise our lesson. PCR, the three steps. The first step, that is the denaturation. Denaturation, annealing and extension. Three phases. We already know this one. Denaturation, primer annealing and extension. So first the denaturation, the double-stranded DNA converted into the single-stranded DNA. Now the primers are going to attach. Then the DNA polymerase or Taq polymerase and deoxyribonucleotides are attached for extension, and in 30 cycles 1 billion of DNA are formed. That is the polymerase chain reaction steps by remembering denaturation, annealing and extension. Denaturation, annealing, extension. The three sequential steps. Now insertion of the DNA into the host cell or organism. So there are several methods of introducing the ligated DNA into recipient cells. Recipient cell can making them competent to receive, take up DNA present in its surrounding. So it is recombinant DNA bearing gene for resistance to an antibiotic, that is ampicillin, is referred to E. coli cells. The host cell becomes transformed into ampicillin resistant cell. If we spread the transformed cell to agar plates containing ampicillin, only transformants will grow. Untransformed recipient cells will die. Those are transformed, they are grow, and untransformed are never going to survive, they are going to die. Since due to ampicillin resistant gene, one is able to select the transformed cell in the presence of ampicillin, the ampicillin resistant gene in this case, that is called the selectable marker. Now the obtaining the foreign gene product. When you insert a piece of alien DNA into the cloning vector and transferred it into bacterial, plant or animal cell, the alien DNA get multiplied. In almost all recombinant technology, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene get expressed under appropriate condition. The expression of foreign gene in host cell involve understanding many technical details. So after having cloned the gene of interest, having optimized condition introduce the expression to the target protein, one has to consider in a large scale. So can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterologous host, it is called recombinant protein. The cell harboring cloned genes of interest may be grown on a small scale in laboratory. This culture may be used by extracting the desired protein and then purifying it by using different separation techniques. These cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while fresh medium is added from other to maintain the cells in their physiologically most active log or exponential phase. The log phase or exponential phase in their growth. So this type of culturing method produces larger biomass, higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce large quantities, the development of bioreactor where large volumes of culture can be processed was required. Thus, bioreactors can be thought as vessels in which raw materials are biologically converted into specific products. Individual enzymes, etc., are using the microbial plant, animal, or human cells. So this is the bioreactors diagram. See the different parts of the bioreactor that provide the optimal condition for achieving the desired product. And by providing the optimal growth condition, every optimal growth condition are provided. The temperature, the pH, the substrate, the salts, the vitamins and oxygen all should be in growth optimal condition. So a stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates can even mixing the oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look the figure closely, see this one. The bioreactor has an agitator system, an oxygen delivery system, and a foam control system. Agitator system, oxygen delivery system, foam control system, a temperature control system, pH control system, and sampling ports so the small volume of the culture can be withdrawn periodically. So now the downstream processing. After completion of biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing or finished product. The process include separation and purification, which are collectively referred to as downstream processing. Very important. Biosynthetic stage, after the all reaction, the product are to be marketed in a finished way, in a finished product that should be presentable. So this is the separation and purification. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials as in case of drugs. Strict quality control testing for each product is also required. That's the quality control testing is very needed. The downstream processing and quality control testing vary from product to product. So what is downstream processing? Keep into in your mind. So we are almost completing our lesson. And this is the mind map that you get your all the revision from this part. So starting from the cutting and joining, isolation, agarose gel and bioreactor, downstream processing, restriction enzyme, cloning vector, plasmids, host competency, bioprocessing, the principles of biotechnology, everything are in a single page for your further information. So everybody are take a screenshot, that will help you. So I think everybody are getting a good revision in this chapter. So we are revise all the segments and important parts from NCERT. So till then, we are moving to our end of the lesson today. So everybody, I think we all present in the next class for the further second lesson of biotechnology. Till then, goodbye everybody. Good night.